Dushka. So our first speaker, we're very excited to have Dino de Carlo. Dino received his BS in bioengineering from Berkeley in 2002 and a PhD in bioengineering from Berkeley and UCSF in 2006. He conducted postdoc studies in the Center for Engineering and Medicine at Harvard and has been on the faculty of the Department of Bioengineering at UCLA since 2008. His research pioneered the use of inertial fluid dynamic effects for the control, separation, and analysis of cells in microfluidic devices. His recent work extends into numerous other fields of biomedicine and biotechnology, including lab-on-a-particle technologies, rapid diagnostics, mechanomedicine, and next-generation biomaterials. He has also been a leader in technology entrepreneurship, co-founding several companies that are commercializing technology developed in his lab, including Cytovel, Vortex Biosciences, Tempo Therapeutics, Foresight, Ferrologics, HANA, and Partillion. He has received numerous career honors, including the Presidential Early Career Award for Scientists and Engineers. So let's all welcome Dino.
Thanks so much, Adam, for the introduction, and thanks for the invitation to come share some thoughts that I have about the future of microfluidics. It's always it's an interesting topic because thinking 10 years in the future is somewhat of a gamble, given that if you told me a year ago that I would be giving this Zoom to all these folks across the world, it seems like. On this topic, and I had a meeting yesterday that was supposed to be in Hawaii, and also had a Zoom meeting associated with that. If you would have told me that a year ago, I would not have believed you. Right? So obviously things change quickly and sometimes dramatically, but I'll try to give a shot at what I'm thinking about, and of course take this as my personal opinions based on my experience, and not and of course I'm sure there's many other thoughts and opinions in this area. So with that, I just have a few slides to kind of anchor some discussion and thoughts, and would love to also engage and have questions too. So let me share this. Yeah, so I've shown this image actually 5 years ago, and at that time I was asked also to opine on what would happen in the future with microfluidics, and there were some topics that I was right about and some that I was wrong about. But the reason I like this is it kind of gives a sense of this pioneering attitude that the microfluidics field has had, where there's some things that we're doing now and we're part of the community, and then there's some things off in the distance, right, that you can kind of see it looks promising, maybe we should go in those directions. And so this is where we can start to think about some of the future directions of microfluidics. And the way that I think about this is in context of where the general the general problems in society are, especially associated with where microfluidics has a lot of impact, which is in biology, right? And if you think about what microfluidics is really good at, it's at automating biology. And you think about kind of the ways that we automate biology and automating biological experiments. Often this starts with things like multi-well plates and robotic liquid handlers. So if you've been to any kind of lab automation meeting, this is really what a lot of pharma is doing right now, is right. They do their biology experiments in these types of formats with well plates. Then you have then you have things like flow cytometers, and then you have things like microfluidics, and you start to get closer to the scale of the biology itself, right? Where here, the scale, the vessels in which you're doing biology and the amount of different experiments that you can do are limited by kind of this form factor. And microfluidics has been one approach that people have been getting around this approach, this form factor, and potentially doing more experiments, either using droplets or microwells or numerous other arrayed approaches. And the nice thing here is that you're also operating usually at the length scale of biology, whether it's mammalian cells or bacteria, or potentially single molecules that you want to analyze. And so this is certainly where microfluidics plays well. And I think the future will be in continuing to look at these areas. One area that I've been spending a lot of time with lately, which also operates really at this length scale, is what we call lab on a particle technology, which also enables interfacing at the scales of biology, single cells and single molecules, and creating compartments at that scale, so that you can start to, as you make compartments that are smaller, you can start to analyze these smaller entities at high sensitivity. So this is kind of the general way that I think about where microfluidics plays. But then we have to look at what's happening in biology. So what are the kind of key drivers of innovation in biology and medicine at this point in time, and how will those evolve over the next 10 years? So the way that I think about that are first on therapeutic side and then the diagnostic side. And so on the therapeutic side, these are kind of the three drivers that you see, in my opinion, of what's driving medicine in the future. And I call these the three pillars of medicine. So molecular therapies, gene therapies, and cell therapies. And certainly most of the pharmaceutical industry has been focused on small molecules, right? So like this verinostat and HDAC2 inhibitor. This is interacting, I just can't find it here, probably here, this small molecule interacting with this protein. And that's been the large chunk of the pharmaceutical industry in the past. And then you start to see, over the last few decades, biologics coming to play. And now I think the majority of new drugs, I think the latest data, are based off of, the majority of the blockbuster drugs are based off of these types of biologics, like antibodies or other recombinant products. Could be mini bodies or other types of molecules that are produced by cells. And so this is another area that you're seeing that will expand further. And then we see that there's a lot of excitement, obviously, in gene therapies. We love CRISPR. We love our Nobel Prize winners at Berkeley. So this is definitely an area where we're seeing excitement. We see some of the first gene therapies that are FDA approved in the last years or so, last year, I think, with Spark Therapeutics. And so this is definitely what will be an area that will continue to grow as we academics and the industry are solving some of the challenges that with delivery, which has been an issue in the past. And then the third pillar is cell therapies. And so we've obviously seen in the last few years huge success with chimeric antigen receptor on T cells. We're seeing more engineered cells, drug secreting cells, microbiome engineering, and then vaccines. I kind of put vaccines somewhere in these cell therapies because you're actually engineering, through some external process, you're engineering your B cells within and T cells to some extent within the body itself to produce, the therapeutics of interest. So, these are areas that are going to continue to grow, cell therapies and gene therapies in particular. And I think these, biologic based therapies, antibody-based therapies as well. And so where does microfluidics help in automating either the discovery of these types of therapeutics or in, the organ on a chip models to study their effects? Where does microfluidics help in delivering genes or evaluating new guide RNAs and what's the best guide RNAs for a particular edit? Where does microfluidics play a role in engineering cells in transfection of cells? Where does microfluidics play a role in studying and selecting and separating out populations of cells for these cell therapies, et cetera? So that's how I kind of envision, think about this. Microfluidics is a tool, it's an automation tool, and it has to be connected to where the field is going, where medicine is going. So I want to also kind of, before I talk about diagnostics, kind of think through some of where it has been in the past. Where has microfluidics been in the past, and what companies and areas are ripe for the future connected to these ideas I talked about previously. And this is how I've broken it down in kind of the decades, and certainly I've missed companies here and I've missed technology. So you can remind me of things that I've missed. But these are some examples. So in the early in the 90s, you had a few companies like Caliper, HandyLab, or Cepheid. The focus was on electrophoresis often, or maybe some automation. And it was focused on molecular diagnostics and research tools. In the 2000s, compartmentalization started, droplets and microwells started to play a role in valve systems, electrowetting and optoelectronic tweezers. We have some favorites maybe for our Bay Area folks like Berkeley Lights, Fluidigm, Rain Dance, Quantalife, Advanced Liquid Logic, Quanterix. So all these are kind of 2000s based companies. And they're leveraging some of these basic fundamentals and addressing markets and research tools, molecular analysis, and sample preparation. Sample preparation and Advanced Liquid Logic was acquired by Illumina to do some sample preparation for their DNA libraries, but I think unfortunately they dropped that project. Maybe somebody in the audience can tell us more why that happened. Third generation, I think we saw a lot of, in the 2010s in the academic environment, we still saw a lot of compartmentalization work. We saw organ on a chip work. And certainly some of the work that we're doing, I would be remiss to not include here, and inertial microfluidics. And then some of the players that were building upon these fundamentals are companies like Cytovale and Micromedicine on inertial microfluidics, organ on a chip companies like CellAsic or Emulate or Heprogen, compartmentalization companies, Zephyrus, Hi5 Bio, Abcelera, 10x Genomics, et cetera, Quantum Matrix. So this generation started to look at different problems like cell sorting or single cell analysis, drug discovery problems. So what's going to happen in the 2020s? Certainly I will probably be completely wrong, but let's suggest some thoughts here. And this is based on some of the directions in cell therapy manufacture, which is a big area of interest now, using things like flow stresses and channels, using microparticles and hydrogels is another kind of core technology, and capillarity, which we've known about for a long time, but it's being used quite heavily now in some of the diagnostic technologies. So you see new commercial players that are connecting to the at-home and on-body diagnostic space. So it's interesting, right? You had some of these point-of-care companies in the early 90s or in the 90s, and then over time now we're seeing, and really amplified by the pandemic, that we're getting towards at-home diagnostics. And by at-home diagnostics, I don't mean glucose sensing, I don't mean pregnancy tests, I mean molecular diagnostics at the home. And so we saw with Lucira, the first emergency use authorization by the FDA for an at-home molecular diagnostic. So that was really exciting because that opens the floodgates, right? So now we can probably have lots of these at-home molecular diagnostics. And certainly there are other companies like Q Health and others that are making technologies that are suited for at-home diagnostics. Here's, like I can show you, here's one of the Q Health systems. You can see how small these type of form factors for these systems. And then you have companies that are focused on cell manufacturing. So I just named a few here: Indie, Selfie, Squeeze, that are focused on using flow stresses to transfect cells in very simple ways, without using lentiviral approaches, without using some molecular approaches that could damage cells. So I think that this is an interesting area. You're seeing in general interest from using microfluidics as a high throughput approach to basically manipulate cells in a very maybe uniform way to introduce genetic material or RNA into the cell, or proteins in the case of maybe a Cas9 enzyme. And then a few other companies here. Partillion is a company that we just started, and Slingshot is another company that's making basically really precision hydrogel particles as building blocks for either automation of life science work or as models for life science like cells and for cell therapy type standardization. So here's some thoughts on what might happen in the 2020s, and I'm sure there will be even more things. But I'm very excited by the, I think when I first kind of joined the microfluidics community in the early 2000s, late 1990s, this was always, this at-home diagnostics was kind of like the holy grail. Can we ever achieve molecular diagnostics at the home? And I think we're seeing that happen before our eyes now. And where does microfluidics fit in this diagnostic space? And so I kind of show this paper that was published recently in New England Journal of Medicine as an example of where we need microfluidics, because you can look at, this particular work is based off of like a CRISPR-based diagnostic for SARS-CoV-2 nucleic acids. But you can see that there's these tons of steps that a user has to perform. So even after a swabbing, they have to like pull down with a magnet, mix the swab in here, pull down with a magnet, take some fluid out, pipette other fluid in, put it into a heater, basically do a bunch of steps and then put it onto a paper to read it, and then take a picture of the paper, et cetera. So certainly this is not at-home friendly test at the current time. It may be eventually, and they might figure it all out. But this is where you really, sample preparation, the automation of these steps is critical to make this something that can be done at home. And this is where I think microfluidics has a role to play in the next 10 years as well. And it's starting to happen. So here are the CLIA-waived COVID-19 molecular diagnostic tests that are authorized by the FDA. I think this is the latest list I've checked yesterday, unless there's been a new approval since yesterday. And so you can see there's not a huge number, right? This is a handful, right? So there's 8 total that are CLIA waived. That means that they're low complexity. And there's only 1 test so far that has been approved for use at home. Of course, it requires a prescription, but this Lucira Health test, actually with some of our alums from Berkeley Bioengineering, Frankie Myers and John Valdisa, and I think were involved in this company as well. So go Berkeley, go Bears, right? Amy, right? Go Bears. Anyway, I guess I think some of these other example tests are I think go Bears, exactly. So I think these other tests, some of them, what you'll find if you read their instructions for use, is they're probably not ready for at-home use. They have a lot of steps. I'll show you one of them, the Abbott Diagnostics ID Now. It's really not, it is CLIA waived, but it's not something that a person can use at home. Some of the tests are more likely to be able to be used at home once approved by the FDA, like the Q Health test, for example. And now here's an example. So here's the Abbott ID Now. And this is the instructions for use. There's a lot of instructions. So you have to basically mix for 10 seconds. You put your swab in there. You press the swab bead head against the side of the sample receiver to remove excess liquid. And then you got to just guard that in a biohazard waste container, which most people don't have at home. You know, and so basically, you know, there's many steps. You have to move things between different places. And, you know, certainly this can be done maybe in a lab, but it's not an at-home friendly test. So there is needs for microfluidics still to make an impact here. And I'm not saying anything negative about Abbott's test. I think that you know, the test works well. It's just wasn't designed for use at home. Or, you know, or by kind of untrained users. So, you know, I have a conflict of interest in Q Health, but I think this is a beautiful example of where the microfluidics enabled them to do something which allows just a single insertion of a swab, and then the test runs, right? The microfluidics enables, you know, valves operate, fluid moves, and the test runs, and you get a result to your cell phone. So there's no, you know, mixing of your swab into a sample, pipetting things. There's no, you know, no like, you know, counting out time, 10 seconds or things like this, which is really not going to make it compatible with at-home use. So here's an example of, I think, an approach that, you know, leverages the microfluidics well, and I think that other approaches can do similar things, and where microfluidics can play a role also in the next 10 years to bring a variety of molecular diagnostics or more complex lab tests to an at-home environment. Okay, so with that, I think I'm out of time, and I'm happy to take questions, or we can take questions at the end. I'm not sure the format exactly.
So let's take 2 questions now. You can type them in the chat, and then we'll unmute you to ask them out loud.
Or just let us know you have a question and we'll unmute you. I guess I can ask one question about the kind of at-home diagnostics. There's a couple of different ways you could imagine someone having at-home diagnostics. You could have like a box that then runs a bunch of tests, or very inexpensive things that you could buy for one-off tests. Do you have any thoughts into how feasible either of those solutions would be in the near future?
Yeah, so I think one of the real excitements about at-home testing is the linkage of the data that's collected to a larger network of health data. And whether that's like helping folks during a pandemic to know, you know, all the people that were exposed to me, or whether that's in the long run, really just building a data set that allows us to have predictive medicine, right? And the more people that are involved in collecting that data, the better, right? And so in that respect, I think that there has to be some component that is a electronic piece that connects to our internet, you know, connected reality, right? So I think that if it's just like, okay, you read something and now you have to input that into something else, that's not seamless enough. And in order to really take advantage of what at-home diagnostics can achieve in the long term, you know, 10, 20 years from now, then I think it has to be connected. And that means you need some type of electronics associated with it.
So questions in the chat. Well, Sean Wang go first.
Yeah, okay. Tell me the order in which I should answer.
So Sean Wang asked his question.
Okay.
Yes, actually, I just typed my question here, so maybe I can just reiterate my question here. So in your last 2 slides, Professor Carlo, you mentioned, I think one of the directions is trying to have the microfluidic applications for sample preparation purpose. So my question here is, are you envisioning more like a general like microfluidic solution to address all kinds of sample preparation, or more like each product or application will need its own associated microfluidic product?
That's a good question. And so, this interchangeability of parts, I think that's always been a dream of the microfluidics community. And it hasn't worked very well, honestly. And it's because each test is slightly different. And it's not like you're processing information on a computer where, there are standards that can be transferred and it becomes easy to make modular components. I think that still there are fundamental unit operations that are performed for sample preparation that it's work, that the academic research enables and demonstrates that then companies build off of. But I don't think it's like they just can plug that directly into their products. I think there's definitely more engineering that has to go into that, but they can build off of those basic concepts. And I think that what you see in the products that are out there now, like these COVID CLIA wave products, is that they do build on basic things that the microfluidics community generated over the last 15 years or so. And so that I think all the folks that have been working in that community, that you could say, be proud of yourself for building all that value. Maybe you didn't get royalties from some of it, but at least it helped kind of get those products out there and being used.
Mm-hmm. Okay, thank you.
So I think Amy had a question in the chat. Yeah, so I think, Amy's question is there non-bio-related fourth wave commercialization applications or a role of big data sciences in the fourth wave? So yeah, so I think that's a great point. So one other aspect of like where we see like the large scale shifts in our society and in science in general is big data, AI, machine learning, kind of those kind of areas. And I think that, yeah, I think that has to be connected to all of these. And I think that's where like some of the home diagnostics areas or other wearable diagnostics areas that connects the most in my opinion, because that's one of the, I mean, drug discovery is another big area where you get a lot of connection to big data. But yeah, definitely the data, automation and data collection are like hand in hand on this wheel of creation of new value, right? Because I think that and you even I've talked to there's venture capital firms that invest in data science or in data companies, and they're very interested in automation companies too, right? Because the automation companies, you're probably familiar with Baidu Ventures, because I think they invested in one of the companies that you founded, Amy, recently. Yeah, so they are very interested in automation because that generates a lot of data, right? And that data can then feed into these algorithms to create value. So that value might be in helping predict health or predict which drug is best for a person, like a more precision medicine side of things, or it can be helping to predict which drug itself is going to bind with that particular receptor, right? So more on the kind of predictive drug discovery side of things. There's companies like Atomwise or other companies that are using these types of approaches to and then you see automation being used in drug discovery more and more. But I think microfluidics has a big role to play there as well. DNA encoded libraries is another direction I didn't talk about, but you have a few companies that are working on automating the DNA encoded library space for drug discovery as well, using microfluidics. But I didn't answer your first question, which is about non-bio related things, because I don't know.
Yardan, would you like to introduce Don? Thank you very much again.
More questions in there.
We're going to move on to Don's talk, and then
okay.
When we get to the discussion, we will finish up with the questions.
Sounds good. Thank you, everyone.
Thank you very much.
And thank you, Dino, again. Our next speaker is Dr. Don Arnold. And for people who have been to our past events, and so you are probably very familiar with Don, who was our stellar moderator for panel discussion for many events. But Don is also an industrial veteran and expert in microfluidics and also entrepreneur by himself. So we're very excited today that he's shifting his role as a speaker to talk about his perspective and his insights about the microfluidics for the next 10 years. So I'm going to introduce Don, reading his bio. And so Don Arnold received his bachelor degree in chemistry from Cornell University and his PhD from UC Berkeley, and studying chemical reaction dynamics and molecular spectroscopy. From 1997 to 2000, Don worked as a senior technical staff position at the Sandia National Lab, where he changed his focus to the development of microfluidics and the microscale chemical analysis system. In May 2000, Don co-founded a spin-out company based on this technology, Oxygen Technologies, acquired by AB Synex in February 2010. In August 2012, he became VP of R&D and principal scientist in AB Synex. Don transitioned to business development in 2015, identifying technologies showing potential for the Synex pipeline. In 2017, Don founded Veristat, where he provides technical and business consulting for a life science company as they navigate from startup to exit. Another note, Don is also on our advisory board. And so for people who are in this community, you're probably very familiar with Don, and now welcome Don to talk about what's going to happen, from his opinion, what's going to happen in microfluidics in 10 years.
Okay, all right, so hopefully I am unmuted and you can see my screen. Thanks to Thomas and Yatian and the team for asking me to give this talk. It's a little weird being on the other side of the Q&A sessions, but should be fun. I very much enjoyed Dino's talk, and hopefully this will be a little bit nice complementary type of discussion. So I started working on this, realized that, like Dino said, predicting out the next 10 years is a little bit of a daunting task when sometimes can't figure out what's coming up in the next 10 days. And so I gave it a few subtitles here, a 15-minute summary of the next decade, or maybe a peek into the micro crystal ball from a commercial perspective, or follow skepsis. I'll let you decide what that means and which one you think it is at the end. So as I go through this, we'll look into crystal balls and we'll see kind of what we think is coming up into the future. You have to start with where we came from, and this will be really brief because everybody's kind of seen some of this. But microfluidics started a long time ago, over across the bay at Stanford, Yaltown and Juan's home territory. I saw Juan out there. The original GC on a chip came about in pre-1980, so quite a long time ago. And things were happening in late 80s, but one of the sexy titles came out of Widmer and Mont's group there with Jet Harrison and Sabath and others, talking about integrating entirely total microchemical analysis systems, and started entire conferences around that. And it's pretty exciting. And just after that, it just kind of started building up. You'd see new technologies and you'd see new companies popping up all over the place. And it was an exciting time because a lot of new things were happening, new fabrication techniques, new platforms, new ways of integrating. And some of these companies are still around doing quite well. Some have been acquired, some have gone off into the history books. But today, we have more than 300 companies involved in microfluidics, so we've really come a very, very long way since the fabulous Stanford GC. So where are we starting right now? I'd say that you have to look at where we are before we move on. And we have a really solid base. We've had more than 30 years of people developing microfluidics technologies that really span a large range of capabilities. Unlike those early days, now we have a pretty good deal of market success. The early days, it was you had a lot of challenges in terms of trying to change people's workflow as well as getting them a new piece of technology. And with some clear examples of success, those are much lower. And we've seen companies grow to large companies and other companies being acquired. So that's given us that validation that microfluidics is real and it's not just a laboratory bench tool experiment. And from the commercialization standpoint, I think it's important that now there are lots of options in terms of getting these parts manufactured. Everything's not coming out of a university fab. There are companies that are stable. They have some fabrication abilities that are pretty broad, and there are different ones who can work with you all the way from prototyping to high volume manufacturing and the various materials that are used for that. So it's a good starting point to see what does the future hold. We have a good set of tools in our toolkit at this point. So the question is, where do we go from here? And that's where it got a little tricky, right? So you say, well, from a commercial standpoint, what are the drivers for the next round of some more commercial microfluidic products? And which applications can microfluidics enable in a way that makes it faster, better, and cheaper, or cheaper? One will do at this point. And then, you're really looking for order of magnitude improvements or maybe enabling entirely new opportunities. Let's see here. So knowing that I didn't know all the answers, I thought we'd ask a microfluidic futurist and found this guy. And I'll ask him some of these questions there. So, the first, he kind of gave me these pretty standard business type answers, right? Like the next decade's growth, it'll be driven by the increasing need, right? You have to have a market you have to have a problem to solve. And, the existing inventions, as you think about this, there are a lot of cool tools. The innovation comes in figuring out how to apply them in a way that solves problems in a marketplace and adapt to changes in the marketplace and the applications. And so that's really going to have to be the fundamental driver. But I think as I thought through this, there's so many things on the outside that are potentially big accelerators. That's kind of where you can find some pretty interesting topic of conversation. And Dino mentioned a couple of these. A couple came up in the questions, so we'll keep pushing ahead here. I think one of the things that's happening is 20 years ago, a lot of work was being done. A lot of patents were put in place, and after 20 years, those patents start to expire. And so I think that's going to be an interesting development that might accelerate some development over the next 10 years. Another one is you're seeing increasing integration into platforms. It was really difficult in earlier times, and the increased fabrication capabilities. But I think the biggest are really some of these things at the bottom. There's a lot of other stuff going around outside of microfluidics that are disruptive, and there's some that were mentioned already: the CRISPR, the new sequencing tools, exploration of the microbiome, other subcellular things that are going on out there. And over the next 10 years with these particular tools, who knows what other dark matter discoveries we're going to make, as I like to call them. We touched on things like AI and machine learning. I think the computational power over the next 10 years is going to expand dramatically. You look at new material developments, the recent implementations here in terms of nanotubes and graphene and microfluidics are going to be pretty dramatic in terms of opening up more electronics-based type of systems, and that'll be big. And the last one in the bottom corner, I agree with here, the COVID response, just a massive infusion of money into that space. is going to have an incredible effect on technology acceleration. It's a lot like why we're all sitting here on Zoom right now. And I've seen multiple projects that have been driven from what would typically be a 2 to 3 year project into a 6-month acceleration, money thrown at problems and throwing out thousands of parts to clinical trials within a 4-month period. It's fascinating to watch. So it's going to be a fun time ahead. If you look at it from a more of a market type assessment, spoke with the folks over at Yola. They loaned me a few slides here, cleaned them up for me a bit. What Yola sees is primarily growth in the life science, and that's consistent with what we heard before. Clinical, point of care diagnostics, pharma being the largest you see on the chart there. Fairly significant market. What they have is a 14% CAGR over the next 5 years in their plot. Assuming that we're to continue, that would indicate that you have a 4-fold increase in the size of the market over the next 10 years. And that assumes that you have linear growth. And I think I'm going to come back to that in a bit. The other way to look at this is to follow the money. Where's the investment going right now? And so they've done some work on that. I don't know if it's fully complete, and I don't have the background behind all this, but leading investment areas right now are shown in this plot. And I think it's interesting that the past 20 months, $4 billion have been raised by companies in areas where sequencing is kind of the leading area, with other genomics. I kind of put them in the same bucket: single cell analysis and then diagnostics. So it's we're consistent, and what we're seeing here is what's big right now. And in the future, it looks like. Before I get a hold of the Yola report, I was making my list of where things are, and I see life sciences clearly at the top. One that gets tossed over into another category that's huge and is microfluidics is inkjets. It's also in the tens of billions of dollars, and it's still growing. And the technology there got leveraged over into other dispensing, and that includes acoustic, piezo, and thermal. There is still a good amount of work going on in actuation, a fairly significant work in liquid lenses for small cameras, stuff like that. Microelectronics cooling, it's kind of come in waves. There's still a push on that, and as the computing continues to go, that'll be a continuing pressure point that may be solved with some microfluidics. The others are further down, chemical synthesis, material characterization, integrating microflow channels with different types of optical techniques. I think the droplet-based work is going to be really, really impactful, not just in doing diagnostics, but also in things like manufacturing. You think of mayonnaise as an emulsion, but you can get really controlled emulsions, and there are already nice products on the market. And then lastly, I put DNA storage in there for a little bit of fun. Life sciences, I won't go through all of this. We've seen there's a huge range of space where life science can work. We've talked about diagnostics, new things coming down. Synthetic biology, I feel, is going to have a huge impact. It can have a huge impact upon it with the microfluidics, or vice versa, driving it maybe symbiotically going there. The role of combination of CRISPR sequencing and these tools, directed evolution coming into and cell-based analyses going into the drug discovery and development acceleration is going to be pretty significant, I think. If we look at I like this plot that we borrowed from Yola as well. They looked at what are the most promising applications in the microfluidics market that haven't yet reached their full potential. And I like the time scale at the bottom, right? We're looking at 5 years over to the left, and 25, 30 years plus out there for those. And what it's kind of telling us is that, we feel like we've come a long ways, but when they look at it, you're seeing most of the markets are fairly nascent stages, and there's a long ways to go before you reach maturity. And even haven't even gotten to that early commercial success stage, in their opinion, right? So 10 years out before we even get to the beginning of their organ on a chip and liquid biopsy. Now, the liquid biopsy, the error bars are pretty large on those dollar values, somewhere between 10 million and 100 billion. I'm not sure about that, but the important point, even with the growth in the next 10 years, there's a lot more out beyond that for microfluidics to grow. If we look at the fluidics to watch, I'm not going to read through these, but I think in working through this, the ones that'll have an impact. you know, these are things that are going on now. You know, things like like I said, CRISPR and microbiome, this optofluidic manipulation. We see a lot of that with Berkeley lights. Droplets, for all sorts of reasons, I think are going to continue to be magical in terms of over the next 10 years. Probably see new waves of tech coming out in pumps, actuators, valves, just to address integration workflows. And kind of seeing some interesting stuff in electrical sensing technologies there. You know, moving away from having to have fully integrated optical systems and such built in. Although those just significantly improved now with you know, the LEDs that are out and available. So anything else there? I'll try to cruise through. Really, a lot of this is to spark ideas. So at the end, we can come back and have a good conversation about this, because I am by no means living in the future. But we can pull words from a futurist here and come back to these outside technological impacts that are coming around. And so Peter Diamandis, he's a futurist. Said, never before in human history have individuals had as much access to capital, computational power, and knowledge. And with the right-minded passion, anyone can solve anything they put their minds to. I like this particular part. Things are converging faster than ever before, and because of this convergence is accelerating in exponential level. And so this is where I come back to this linear plot. You know, it's a nice linear plot, but you know, I think that I don't think it'll remain that way. Just, you have microfluidics on its own, but now as you've got all of these other disruptive technologies on their own that can either benefit from the ability for microfluidics to take workflows down to the molecular or cellular levels and make things change in a way that's not really linear anymore. So I think there's more to it than what's shown in the plot for the next 5 years. And you know, this thought came out as I looked through each one of these individually and considered what would be the impact. Pretty daunting in many ways. Trends. I'll just skip through this pretty quickly. We have some good pictures there, but I see, while early days integration was, you know, the microtasks, but I remember sitting at one of the microtasks meetings and Jed Harrison had thrown out the concept that, you know, integrate only where it makes sense, because otherwise you're making things overly complicated. And I likened it, when I talk to people about this, it's a bit like, you know, when you change your oil in your car, you don't necessarily change the transmission at the same time. So why would you have an integrated detection, you know, $1,000 detection system on a $5 chip? But I think that what's happening with the manufacturing and the external components is that the cost of manufacturing integration is coming down to the point where you'll see an increased amount of workflow automation and integration into these systems. That with the improving in the computational and the algorithms and cloud-based data storage, closed loop and autonomous operation or monitoring systems will end up being dependent upon these. Multi-mode detection, and as Dino was talking about, the penetration of the consumer market there, you know, the home-based diagnostics, phone attachments, and as I thought about this when I said, what's a phone going to look like in 10 years? For goodness sake, it was 20 years ago there weren't really cell phones, so now that's the phone kind of is your main addiction in life. And then the last one here is manufacturing. So I think a lot of this being driven by droplets, being driven by closed loop and drug optimization. The concept of having droplets and droplets to give you controlled drug release, or having active ingredients that will react as the molecule dissolves in situ next to a cancer cell are fascinating. And I think those types of things will be enabled by microfluidics. And you can see here some, the emulate chip here, actually being able to see with human tissue could lead to improved drug development, could be personalized medicine, or somebody has their own emulate box, they build it up, put your emulate chips, figure out what drugs work for you. Directed evolution, I think, is just going to have an impact on everything, and those can be run within a microfluidic system. The power of that is amazing. You ever see a Berkeley lights there? They can, just by using optofluidic manipulation, scan thousands of cells looking for the absolute best one that's been coming out of a whole bunch of different genetic mutations. Here, I can't remember where I even got this one. The whole system just clips on and uses the camera as your detector, right? The other part is closed loop. This is, I don't know if anybody's familiar with the cyclofluidic. Dave Perry was there for a bit, and it started at GSK Pfizer, came out, and then they eventually ended up closing up shop. The idea was to have a fully closed loop drug development system at a micro scale. And I was fortunate to be able to work in early days on some of that. Very, very exciting, maybe a little bit early, but I think, as over the next 10 years, the benefits that would come out of something like that won't get lost. And then the personal space, closed loop diabetes systems have just come out, and those are not necessarily microfluidic at this point, but I could envision that becoming the case very easily. Noel's perspective is looking at this from having a multimodal testing platform. So rather than having 5 different platforms on which you do this, now you've got microfluidics enabling you to run on one platform a whole bunch of different tests simultaneously. And so that drops your cost. It drops a lot of it adds a lot of value to the person being tested. What about the materials in the fab? I think I won't spend too much here. This is the old picture of this. Looking forward, I think you'll see much more in the world of hybrid combinations using kind of existing materials. PDMS, I'm not a huge fan at all, but I see that it's very popular for prototype materials and has real applications in products. I don't think that once you get to the high volumes, it has some some issues that people get around and then transition over. For, as I mentioned before, I think more electronics-based devices, we're seeing things with of course nanopores and FETs are enabling kind of mind-boggling achievements nowadays. The other thing out there, we had Albert Fulch speak a little while back, additive manufacturing. I was a skeptic, but I'm seeing new things that are coming out that actually seem like certainly for accelerating development, the additive manufacturing and some of the cool stuff out of Eastern Europe with the laser-based microfabrication of microfluidic devices is really exciting. So Albert had great polymeric, and they had some ceramic-based materials out of those femto-based systems. And then, the question is, what do we do? We're going to be generating more plastic. This came out of a conversation with Yolanda a couple of days ago. What do we do with all the extra plastics? Maybe some people will go in and try to develop some green manufacturing techniques for these. Path to market. I feel like I'm probably getting close on time. Tech dev is going to continue at the normal places. Commercialization. Startups, they kind of have limited money and time, so they have a few options: either grow or get acquired or disappear. And the organic growth within large corps, not necessarily as innovative as what happens in the startups. So they end up doing acquisitions, and they either leave it alone or they start acquiring and bringing in companies and then putting them all together on platforms. And so interesting right now, the largest microfluidics company in life sciences are Illumina, very much an organic, and bioMérieux, same thing. But Abbott, Danaher, and Thermo, significant acquisitions of microfluidic plays in recent times. I found an interesting note. Some companies we looked at several years ago. Scandinavian Microbiome Devices, one of my favorite companies for a while, and Abaxis. We're moving down the path of actually wonderful microfluidic devices. The idea was to risk mitigate by penetrating the vet market first, veterinarian medicine market first, diagnostics, and then move into humans. But we can see is Zoetis actually now owns both of these companies. Scandinavian was purchased for $80 million, and Abaxis for $2 billion effectively. So, so Zoetis has a pretty interesting portfolio out there. It'll be interesting to see where they go with that and how many people move through that trajectory over time. Then, of course, there's the way out there stuff, and that gives me a sign I'm getting close to the end. So there's data storage. DNA storage is something that people have spoken about for a bit. I think in order for us to hit those 20-year out targets, things will start to happen over the next short while. And there is definitely motivation, right? 455 exabytes of data per gram for DNA you can store. If you think about the entire internet, check this today. Today they have an update scroller thing going on. The entire internet is about 18,000 exabytes. So you need about, no, that's not true. It's a little bit more than 41 grams. I got that off by a little bit. But you can imagine there's not a huge amount of DNA required to hold the entire DNA there. Type that in properly. For personal health monitors, you know, we saw the tricorder back in Star Trek days, tricorder 1, and then as Star Trek went along, we got the new one. This was a smaller single handheld unit. I don't know if they had microfluidics in there. It's, you know, futuristic, of course. As things go with more and more integration, we'll see these not necessarily things like this, but the diagnostic parts coming through. But I think, you know, perhaps you'll end up getting in with the nanorobotics. You know, maybe you'll get into the nanorobus. You know, that you guys recognize this. These are from Star Trek and from Magic School Bus, where they went down and there's a whole bunch of kids in a bus and explored around. But perhaps you'll have, you know, I think that, you know, some version of what Theranos was trying to do will come back. Probably not everything, but some variant of it will make its way to market within the next 10 years. I doubt it's going to be a little bus riding around down inside the nanotainer, though. And lastly, but of course not least, space travel. So I decided, well, in this case, I can't use the little water guy. I need a new crystal ball. So let me recognize Hal from this, you know, 2001 Space Odyssey, but he wasn't very helpful. Yeah, I'm sorry, Dave. But I disagreed with him, because if you listen to Elon Musk, he's planning to have a colony on Mars in 2027. And so, of course, that's less than 10 years from now. And no one wants to be hauling up a Waters LC system or a SciEx mass spectrometer onto Mars to be able to do their analysis of what's going on. So I think opportunities for microfluidic analytical systems, they've already been to Mars on other rover systems. They're going out to moons around Jupiter. You know, that's a place where it'll go. Before it was onesie twosie. You know, if they have a whole colony up there, there may be more. And of course, just to get there, it's going to be a 9-month journey. So monitoring stuff inside the system is going to be important. And if you want to test it out, you know, one good place, it's not outer space, but it's out in the ocean. So maybe some of these things can be test marketed on cruise ships. So, okay, a little bit of entertainment. Oh, I forgot about this. So oddly enough, when started I think Yantian mentioned I switched from chemical physics to microfluidics. When I moved to microfluidics, this is the box we were building at Sandia National Laboratories. We called it the Microchem Lab. Oddly enough, it looks a lot like Bones's little tricorder there. So let's see here. What's next? All right. So to wrap this up, you know, my feeling here, and it's a purely a personal attempt to generate thought and conversation. The future of microfluidics is bright. It's nonlinear, and it's getting smaller every day. And at this point, I'd like to thank you for your attention and your patience. And I want to hear what you guys have to say. And lastly, I found some red and green droplets over on a digital microfluidics thing. This cat looks a lot like a cat that we adopted during COVID. So I thought that was kind of cool. And then, of course, here's one of our favorite crystal balls moving into the holiday season. So, thanks very much, guys.
Thank you very much, Don. That was excellent. So we have lots of ideas to discuss, and in the interest of time and efficiency, we're going to move on to the discussion portion, and then there will be ample time for you to ask questions of both speakers after Yolanda gets us started. So with that, I'd like to introduce Yolanda. So Yolanda Fintschenko is a scientist, writer, marketer, and avid runner who lives and works in Livermore, California. Yolanda holds a BS in chemistry from Trinity University and a PhD in bioanalytical chemistry from the University of Kansas. After a brief postdoc, she began her scientific career in the field of lab on a chip at Sandia National Labs, working on devices for biodetection for biodefense. After 9 years at Sandia, Yolanda moved to industry to build an international marketing team of scientists to exert technical influence in the field of food safety for Thermo Fisher Scientific. Food seems to be a common theme today. 2 years later, she took that expertise to Labsmith to run marketing and sales, While at Labsmith, she began a passionate advocacy of the holistic digital marketing approach tailored to technical products and markets by starting her first company, Common SciSense, in 2015. In 2017, she co-founded 2 digital marketing agencies that focus on business-to-business digital marketing for technical or scientific products, Founder Traction and Fixate.io, and remains co-founder and principal at Founder Traction. So Yolanda, thank you very much for joining us, and we look forward to what all 3 of you have to say.
Well, thank you very much. Thank you so much for inviting me to participate. And thank you, Dino and Don, for your vision of the future, or I had to actually look up Don's word, or navel gazing, whichever you think it might be. So, but sometimes
my favorite words.
To move forward, you need to understand yourself. So all is not lost, even if it is navel gazing. The thing that comes to mind in both of you had very similar visions. And then that was, of course, a lot of that supported by data that you presented, Don, from Yole Development. Looking at these, you know, what's going to be happening, particularly with genomics and personal diagnostics. What do you see, Dino, as the most, speaking, I know you're also an entrepreneur, but maybe you can kind of wear both hats. What do you see as the most important question that needs to be answered, or maybe asked at this point in order to achieve this future state? And then for you, Don, what do you think is the most important technology that needs to be either invented or matured in order to achieve this future state in 10 years?
Yeah, thanks. That's a great question. I think, you know, when we think about like precision medicine or personalized medicine, there are many kind of stages of that. And I think the first stages are kind of already here. You might look at like Guardant or Foundation Medicine or something, you know, many of these companies that are sequencing, let's say, information from tumors, getting particular mutations that are connected to drugs that then target those mutations, let's say. So that level is already kind of in play. And it seems like for the cancer field in general, this idea of kind of sending out things to get tested and then getting information back is probably fine. You don't need this real-time kind of feedback because the disease kind of progresses at a rate that is not that fast. I think that where precision medicine, where it can grow and where there's needs are more in precision medicine with disease states that progress rapidly, and this is where microfluidics, I think, can play a role, right? And so this could be infectious diseases. And we know, even in COVID patients, right, we know that not everyone responds the same way to an infection. And is there a way that through some diagnostic test that you could, whether, that's the pre-existing antibodies that patient has, from serology or some other aspect, you get information about that patient that then helps in treating that patient or in that patient's health journey, right? So I think that's those types of areas we have to think about where microfluidics can play a role, where the disease state is progressing rapidly. It's not something you can send out to a lab and then those labs can automate it in mass, and that becomes much more much more sustainable for those types of models where disease is slow. But in the cases where disease is rapid and there is a genetic component or some microenvironment component, epigenetic component, those are places where microfluidics, I think, can play a bigger role.
Right.
Yeah, and I think from the what needs to be developed, I'll turn it a little bit differently in the sense that I think we have a pretty solid toolbox to get to platforms that would function. And when you get into things like the personalized medicine, when it was first being discussed, I was somewhat skeptical because a lot of the regulatory things that we have in regulatory structure that was in place was really designed to have a therapy that would treat a broad range of people. And then those that were on the outer edges of that curve, it wouldn't work. But that was, you had to make sure that it worked for the majority of the people, which is exactly the opposite of the concept of personalized medicine, where it's designed specifically for you and your particular genetic and proteomic makeup. And the question became, how in the world would you be able to get anything that would be a drug for a particular person through a process that was designed to weed it out? And I think, what's happened is, the sequencing information and all of the diagnostic tools come around that will now allow those. And it looks, I think the biggest fear was trying to figure out how do you navigate through a regulatory system and figure out how do you put together something that could be marketable as a product, use the tools that are available, and then still be safe and effective for patients that are using it. And I think that the other side, even for the slower moving drugs, though, I think some of the things that would be beneficial is monitoring the effectiveness of therapeutics, or monitoring whether or not people are actually taking their medications in situations where that could be a pretty helpful type of tool there.
Great. So again, what I heard, is that you're looking at basically adding value where time is of the essence, where you can use microfluidics to drive towards real time in medical situations that are where that's necessary. And Don, again, coming back to the personal side of the new medicine, is making sure, or innovating in terms of the regulatory frameworks, and then also maybe having to innovate in order to respond to regulatory frameworks, to make sure that with this, this kind of real-time or personal data, that you're able to actually move on these corner cases as quickly as possible from a regular.
Yeah, that's right. I mean, a quick example, the concept that I showed, the closed loop diabetes treatment, there early on in our formation of our company, we had a glucose sensor and we had an insulin delivery pump that were all, the size of a finger. And the concept was to talk to companies about tying the two together in a closed loop and having basically the artificial pancreas. But at that point, there was no way through a regulatory process that anybody would touch it because of liability. And so it just went for years. I mean, that was more than 15 years ago. And now, using traditional mechanical systems and electronics, they've managed to work their way around that, and they have a system where they can close the loop. And I think a lot of it has to do with the data analysis and the algorithms that people have been able to develop over the time to address the risk and manage that. So there are other things besides the actual hardware.
Right. So kind of building on that, Don, so my daughter has type 1 diabetes, so I actually know a lot about the evolution of the improvement of standard of care, especially the artificial pancreas. And one of the biggest drivers, the thing that got the FDA's attention, was the patient community. So one of the reasons this happened suddenly, it would seem like suddenly, was patients, basically, there's a mantra, we are not waiting. And because of GitHub actually developed their own DIY artificial pancreas and started to work on that and distribute that information to patients. And because there was no liability associated with this, because everyone was just building their own. So with that knowledge, I'm asking the question, because there's a strange combination, right, of hyper globalized companies and these huge acquisitions, big pharma, Thermo Fisher acquiring so much technology, but at the same time, with the advent of microfluidics and a lot of computational tools, there's also a democratization that's happening at the same time. So both are true, right? So it can be a highly, in the commercial side, there can be very few players, but at the same time, there's a lot of opportunity for a DIY approach because of things like GitHub and some of these DIY biolabs. How do you see that influencing the futures that you guys presented, and potentially accelerating it, or is it relevant because of the needs for some of the computational power that could be outside what even a DIY motivated programmer could get to?
Yeah. You want to go first, Dino?
Sure. Yeah. I mean, so I think that, yeah, I mean, I think that, yeah, there is this trend towards democratization across fields, right? So I can talk about it in a few different aspects. So democratization in terms of the some of the most sensitive assays, like digital assays for proteins or nucleic acids, or democratization for single cell assays. Right now, that is something you need very expensive equipment for, and so it's hard to build around the infrastructure. Right? You have companies that are selling instruments, and then they sell things that you can't really modify, and then you work with those. Biologists generally are okay with that if it's solving their problems, but often, let's say that now I want to look at an epigenetic thing along with, a genomic thing. And so then they have to work with these companies, and then now they have to pay more to access this. And so I think that's limited some of the innovation. And so I think that there is going to be a move, and we're working on some of these lab on a particle approaches that'll put more power in the hands of the academic researcher to build on that platform and use that platform to do these assays in their own ways without expensive equipment, et cetera. And I think you see that across other areas as well. And then on the diagnostic side, a home diagnostic really is a democratization in some ways, right? You don't have to, especially if there's not a prescription required to run it. You don't have to go to the doctor and say, hey, I feel like I'm sick and I want, and you have to have them say, okay, you need this diagnostic. Of course, people may push back and say, well, the doctors know best, and if you don't need this diagnostic and you don't need this drug, then you shouldn't have it. And I think certainly having a doctor in the loop for medical decisions is really important. But there are cases where that, like you brought up for type 1 diabetes and making these DIY, closed loop systems where, the people know what they're doing, and they are taking the risk in their own hands, and you give more power to them to make those decisions. And I think you'll see that as more of these diagnostics also go into the home settings. And there's a balance obviously there that public health piece of that, but obviously you don't want people prescribing themselves drugs that that they don't need. But certainly it's not like an all or none, I guess I would say. It's like, having the access to diagnostics, we already do it, right? You have glucose sensors, you have pregnancy tests. And so there are some level of these things that are not going to, I think, more power to the patient, the person to make decisions is going to be the new trend, I think.
Mm-hmm. Thanks.
Yeah, I was just going to say that the GitHub concept and the ability to form communities, like all of these incubators that are being run around the country now, and just everything that's available on the internet generally it fits into that category of the stuff outside of microfluidics that's an accelerator, and just bends the curve upward in terms of where we can go in terms of speed. So yeah, I think it's critically important. I neglected to add that to my list.
Thank you.
So cell phones are another big part of that too. Like you had that.
Yeah.
I mean, you can almost walk around with your own lab right now with a few modifications, but yeah.
So I did double check there. I did that DNA storage thing, thinking about the amount of storage on the internet. It is only about 41 grams of DNA, so you could hold the entire internet in your hand if it was stored in DNA.
That's easy.
Team too small.
Too small.
Very efficient.
You know, the crazy thing is we'll get there. That's what the history of technology development has taught us, is if you can calculate it or imagine it, we get there. So, Thomas, I just want to do a time check. Is it time to move on to some questions from the audience?
Yeah, absolutely. You can sprinkle them in, and then if that dies down, you can continue on. So, Adam, do you want to? Where did Adam go?
I'm here. Sure. So we have a couple of questions about kind of the home testing thing, and we touched on this a little bit maybe, but what sorts of readout approaches would be best for home testing, and what are some of the biggest challenges for creating those?
I can start there. So I think I kind of touched on some of this earlier, which is you want to be connected to your cell phone or other data storage and distribution capability to get the most out of the home testing capability. So what that means is that you need a readout that is going to eventually go to electrons, right? So that could be directly to electrons, electrochemistry, or it could be through some optical sensor that then goes to a photodiode or some type of CMOS or et cetera, to then electrons. But yeah, so I think that you don't, the lateral flow readouts can be potentially used, right, where you have a colorimetric thing, but you have something that's reading it out, right, some optical sensor that's reading it out. And you see readers like that that companies are selling. So yeah, I think that there's a multitude of different readouts that could be used as long as you eventually get to electrons that can be, help to really get the power of the distributed aspect and data storage aspect and continuously monitoring aspect and all these aspects that really make it powerful.
Yep, I agree with that, and the ability to move all of the electronic data analysis stuff off into the phone just accelerates commercialization, because the cost of all of those things can be taken out of the product that's down to just an accessory, right? Yeah.
Should I read questions, Thomas?
Sorry, yeah. Adam has compiled a list that he was reading off of.
Oh, okay, got it.
Yeah.
Sorry.
So we had a little bit of discussion in the chat about other applications beyond biology, in particular things like material synthesis, or even, I think Amy was mentioning cosmetics or other things. I was wondering, do you guys have any insight into, maybe not like what applications are immediately available outside of biology, but how people might go about matching microfluidics to an application that is kind of outside the traditional realm?
Yeah, I think you could come back to these basic concepts. What is microfluidics good at doing? It's automation of fluid operations, right? So if you're doing something in a test tube that used to be done in a test tube and then was done in multi-well plates or small little vessels or in continuous flow chemistry, potentially you can do that in using microfluidics. Or maybe with them even, in material synthesis, usually maybe done on a larger scale. So I think that's where you have to, try to understand, is there a benefit of automating, or, I mean, the other aspect that microfluidics allows you to do is there's some unique physics, right? So, and that could be the droplet formation is very precise. And so you see that I think Don touched on this as well, like synthesis of these microparticles or emulsions. Those are areas that you see, that will continue to be work in, and because you have a lot more control than you would using kind of just emulsion formation in a normal reactor. Yeah, so I think that those, you just have to think about where is automation, where is the physics-based control needed, and then that can help tie to the applications.
The chemical synthesis project that I mentioned around the cyclofluidic, the value there wasn't so much in the just the biological scale. It had to do with a thermochemical type scale too. So for example, in the chemical synthesis, the move from a small reaction vessel to a scale up at a pharmaceutical company for a small molecule now, not a biological molecule or anything. So it could be applied to anything. Depending upon the kinetics and the heat transfer required for the reaction, the scale up could be very different from a 100 milliliter flask to a 100-gallon tank, and that would be very challenging. And so the idea was that if you move into a system that's microfluidic, you get very well controlled thermal transfer through devices if you design them properly. A lot of work has been done in Jensen's group at MIT around this. Mix things, you can do things that would typically be super dangerous, just explode on you if you keep them confined in a microchannel. You can do syntheses that aren't really practical on a benchtop. And so those types of things in a microfluidics are kind of different motivations. It's more about using the scaling for other practical purposes in the engineering of chemical synthesis. And that's another driver that's not really that's limited to the life sciences there. Similarly with the microelectronics, cooling was one space that was in that case where you're basically trying to shrink the distances over which heat has to be transferred out of a device that has a very high power heat loss and needs to be cooled effectively.
Thanks. So Aditya had a question about integrating fluidic logic gates for microfluidics and automation. And I would expand that to ask what sorts of features are required to kind of get a highly multiplexed system on a microfluidic chip.
Don, do you want to?
That one looks like it's for Dino and Juan. I see Juan out there. Yeah, I haven't done much fluid logic gates.
So yeah, so I think the fluid, there's only a few folks that are working on kind of microfluidic logic gates at this point that I'm aware of, including like Elliot Hui at UC Irvine, and so the point there is that you have I think this is what you're referring to. You have the computation done in the microfluidics. Is that what your question is about? Like when do you want to do the computation in the fluidics circuit itself? I just want to clarify, is that?
So I'm not quite sure what Aditya was talking about, and he was telling me his microphone was having some issues.
Okay.
Hi, Adam, can you hear me?
Oh, yeah, we can hear you.
Okay. So my point was that basically logic gates can be used for automation. We can do numeric, simple computing purposes. So my point is that maybe we can automate the processes on the microfluidic chip, like turn on the gate and do the mixing in a microreactor, or do the sample prep and turn off certain other gates. Basically like what has been done to transistor logic and FETs. Can we do that with liquid in a microfluidic chip?
Right. So there are approaches to do logic, to have clocks, to basically do computation using pressure pulses, basically, and so that technology has been developed. But for many of like let's say diagnostic application, you can have that computation all done in electronics, and then interface with the fluidics, right? So you can turn on a valve after a certain time as it passed, in your electronic clock. And kind of you do that process, but control it with electronic. So there's some niche cases where that might not be, where having it all in the fluidics might be valuable. I think some of those connect to like high throughput screening. I don't know if you're familiar with like Elliot Twee's work, but one of the challenges with like multiplex valving in microfluidics is all the lines that have to go into that system. And so controlling many valves, a huge valve array is somewhat challenging unless you do some logic in the microfluidics itself. So that might be an area where it's useful, but I think for diagnostics it's less useful because it's a simpler process in essence, right? There's usually a simple workflow that you're doing that's one after the other, and you don't need the microfluidics to do the logic itself. But there may be some reasons that you might want to do that. So if you have some, please let me know.
Well, I actually have a couple more questions for you guys. One of the things I know you both are coming at translating technology from kind of two different places. So I'm curious, Dino, coming from academia, and then Don coming from the commercial side, pulling things into commercialization. What do you see as again, what do you see as either something that needs to be changed, innovated, or accelerated in order to improve the tech transfer process in microfluidics?
It's an interesting question. So the normal tech transfer process from the academic setting often is not thought through from like early stages of the project. And maybe Don has seen this from the other side. Is like, someone in an academic lab is kind of just, playing with things and thinks they know the needs. And so they move that project forward, and at the point at which it's published, and it's like, okay, well, maybe it's time to translate this. It's clear that some of the early assumptions that went into that project were not really relevant to the commercial needs. And so I think that that's something that as academics, we need to do a better job of, is at early stages of a project, even talk to folks that are commercializing, be involved in these types of communities and start to learn, where are the needs? Where are the biggest needs? And how does the work that I'm doing best address those needs rather than make early assumptions and that, oh, I know what's right here. I think being more humble ourselves as academics and saying, oh, well, I need to talk to more people and understand what those needs are. And that can kind of aid in the translation of technologies and tech transfer.
Yeah, I would, I'll agree with that for the most part. When you see, anybody who's gone through it was there at some point, right? They had a technology and trying to figure out what to do with it, unless they came at it from the business side initially. But the idea is, do you figure out what is the problem that needs to be solved, and what does the product look like that would solve that problem, and then what's the technology that's supposed to be inside of it? And it doesn't always go in that direction. Most of the time, you have a technology enthusiast, they have an idea, it's really interesting, but none of the market work has been done yet. And one of the things I'm doing now, separate from the Veristad consulting, is with the institutional co-investor, where effectively we bring experts in that are early in technology development person, wants to explore commercialization, and effectively we pair them with a market expert to help them flesh out what is the real value. Is there a patent? Are there any patent issues? And then from there, assess what it would take to actually move it from what is currently the lab prototype out into a market prototype that you could use to really test the market thesis hypothesis that's been pulled together. And those types of things are forming, the incubators are building these things into their workflows. It's, I think that probably the best thing that could happen is, when people are thinking about taking out technology, find those types of people to help them get that in place before money gets spent on something that, ends up with hurdles that have nothing to do with the technology that really prevent the commercialization. And that's happening a lot more now. It's very, very good to see. But there's always a very, very first set of conversations to have with the new prospective company forming person.
Great. Well, thank you both very much. And I think we're out of time for questions, but Dino and Don, I think, gave really a wonderful vision of not only where we'll be in 10 years, but how we might get there. So thank you very much.
Yeah, I'll give a virtual round of applause to our 3 presenters. So we have one more thing on the program tonight that will be brief. We have 3 job postings this event. So I will pull those up here. So the first one, Thermo Fisher has 4 positions available, and Savat is here to say, give a little 1-minute spiel on that. So I'm just going to unmute Savat.
Thank you, Thomas. Thank you for the discussion and the opportunity, Bay Area Microfluidics Network. So I'm part of the Thermo Fisher Scientific Genetic Sciences Division. And we are essentially in the South San Francisco, Pleasanton, and Santa Clara. We have 3 offices. We are supporting within the Genetic Sciences Division. We are the core platform engineering R&D team. So we are supporting capillary electrophoresis, qPCR, microarray, and molecular diagnostic platforms as a group. And we have quite a bit openings these days, and I wanted to come here today and kind of give you some of the openings that we have in the group, specifically around the microfluidics expertise and also bio engineering and etc. So if you are interested, please let me know. I think my contact information is listed here, and I'll stick around after this job board. And if you want to talk to me, please contact me. Thank you.
Thank you. And I also posted the links to those job postings in the chat. You can also find more information there.
Thanks.
So next up, we have a position from Care and Biosciences. And very briefly, Karen is developing next generation therapeutics that address significant hurdles in the treatment of serious diseases. Karen's proprietary drug discovery platform delivers an exclusive view of uncharted routes to new medicines. Our platform utilizes the dynamic multi-scale cellular adaptations that underpin clinical outcomes, generating multidimensional predictive disease biology datasets, fueling our drug discovery pipeline. So feel free to send me a message and I can give you the full job posting, which we'll also send out in an email post-event. And last one tonight is for a postdoc position in Amy Herr's lab. And again, briefly, applications are invited for a postdoctoral scholar position in the Herr lab at University of California, Berkeley. The scholar will lead research efforts on an NIH-funded research project, the Comprehensive Cancer Center at Stanford, and the Chan Zuckerberg BioHub. The overarching goal of this project area is to design, prototype, optimize, and validate high selectivity microfluidic tools suitable for direct measurement of proteoforms, including protein isoforms and single cells. Dual mode measurements, which were named the #1 method of the year by 2020 Nature Methods, are an important emphasis. And so again, feel free to reach out to Amy, whose email is on the slide, or visit the Her Lab website for more information. So next up, if you are interested, we'll have a networking session, and feel free to stay around as long as you want to chat. Everyone will be unmuted. Please turn your video on if you're able. And in Zoom, you can move around. You can choose which breakout room you want to join. So there would be 4 options, one for each of the speakers. Or if you prefer, the speakers can also join the same breakout room or head out for dinner, whatever you'd like. And participants can then join whichever room they see people they want to talk to in. If you haven't updated Zoom, just send me a message. And with that, I will open up breakout rooms. Again, you won't be asked to join a breakout room. You just get to pick and choose the breakout room of your choice. So thank you all again very much, both presenters and also all the participants who joined this evening. I hope to see you all again soon. Stay in touch and have a great rest of 2020.